It is my great pleasure to introduce our speaker, Tim Wheeler. Tim is coming to us tonight from his home in Arlee, Montana. He is a geologist, mycologist, lichenologist, and photographer who works at the University of Montana as a research technician. And his work there focuses on the genetics and the genomics of the tree of life. Tim has been studying the fungi of Montana for more than 20 years and has more than 16,000 specimens in his various collections of fungi, lichens, and non-vascular plants. And I highly recommend that you take a peek at his website. That's timwheelerphotography.com. And you can see much of his published work as well as his outstanding photography. So welcome, Tim.
Hey, everybody. Thanks for coming out. Can you hear me?
I can hear you great. Thank you. I'll go ahead and mute myself.
How about that? Does that pop up for everybody?
I can,
Yes, very nice.
your screen looks great.
Yes, thank you. Good.
All right. Should we get started then?
Yep, go ahead.
All right. Thanks, everybody, again for coming out. I'm Tim. Thanks for that introduction, Beth. Yeah. So tonight, I'm going to give a talk. I'm going to give an introduction to fungi. We're going to start out kind of at the basics, get everybody on the same page, talk about what a fungus is, what a mushroom is, and some cool features about them. And then we'll go through. That's like the first third of the talk. Then we'll go through some different groups of fungi that are pretty or have interesting stories or edible mushrooms, whatnot, or have cool ecologies. And then I'll kind of wrap up with some other wow kind of facts and stuff like that. So if you have any questions, please type them into the chat and we'll get to them at the end. Okay. So if we start the basics, there are 7 kingdoms of life this week. Next week, there might be 8. The following week might be back to 5, so this is kind of in a state of flux. But what we're going to be talking about are the true fungi. So this is the kingdom of the Mycota. Oftentimes, slime molds get talked about and get grouped in with fungi, but they're not actually true fungi, right? They're protists. So we're not going to be talking about slime molds tonight. If you have any questions, we can try to answer them, but I don't really study slime molds. But yeah, so we're going to spend most of the time in the Mycota. So if we jump into this group, like I said with the kingdoms, up until there may have been 8 phyla up until like 2018, and then in the last 4 or 5 years, this number has ballooned to 25 phyla now. And it's mostly these early diverging microscopic things. And so if we look at this list here, what we're going to be talking about today, like I said, the title is Macrofungi. So these are big fungi, the fungi that you can hold a fruiting body in your hand. And these are in the 2 groups, the phyla Ascomycetes and the Basidiomycetes. And so the Ascomycetes are your cup fungi and most lichens. And then your Basidiomycetes are your mushrooms in other fungi. But these other ones like the Microsporidia, the Cryptomycota, you might have heard of chytrids attacking our frogs and amphibians, the blastoclades, the zoopags, and the mucors. So these are all microscopic fungi that live inside insects, live in the guts of animals, or are quite tiny. So you might never run across these, although many of them are very important. You don't really see them when you're out in the wild. Okay, so here I have a tree here, and on the bottom, this is the fungal tree of life. So this is just the Mycota branch on the tree of life. And on the bottom is the time scale. So we go from 0 back to 1,000 million years, so back to 1 billion years. And you can see, I made this figure 5 years ago and I need to update it, but the origin of fungi as a whole has actually been pushed back to like 1.2, 1.3, even 1.4 billion years ago. And you can see that big green triangle and the big red triangle. Those represent the Ascomycetes and the Basidiomycetes. And then there's some vertical blue bars going up in the background. And I just put those in. These are kind of major Earth history events. So you can see land plants were about 500 million years ago. Some of the oldest lichen fossils are about 400. Dinosaurs around 250. Birds younger than that, and flowers even younger than that. And what I want you to understand from this figure is that fungi as a whole are really old, right? They've been on this earth for over 1 billion years. They figured out how to survive everything the earth has thrown at them. And so some groups of fungi are actually more distantly related than, say, humans are to mice or humans are to insects. So I just want you to keep that in mind, that when you talk about fungi as a whole group, it's quite diverse, okay? And we'll look at a bunch of examples of that tonight. And here's just another schematic, different way to look at the tree. And you can see at the bottom there, archaea and bacteria. Then we branch up to the plants and other kingdoms. And it shows here that fungi are really closely related to animals. And hopefully that's common knowledge now. Historically, they've been treated as plants. And recently we actually got the mycological community voted and agreed that we needed a third F. So we now have what's called funga. So mushrooms are no longer lumped in with the flora of an area. So they have their own F, funga. So it's fauna, flora, and funga. And so as you move up the schematic in the mushroom, you can see some of those groups. And below those group names, there's the number of species that are currently described from each group. So you can see all these microscopic things, not super diverse. That could be an artifact of people not looking for them. And then if you get into the cap of the mushroom here, you can see about 100,000 species of Ascomycetes and about 50,000 species of Basidiomycetes. And these are fungi with a name. And as we'll get to the end of the talk, you'll see there's many fungi that don't have a name yet. And in the top there, you can see a bunch of these group jelly fungi, rusts, lichens, and things like that. So I'm going to kind of walk through some of these groups here in the talk. Okay, so what is a mushroom? One of the easy ways to visualize this, mushrooms are just the fruiting bodies of certain fungi. All right, so one way you can think about it is it's the equivalent of the apple on an apple tree, right? The rest of that tree is the fungal body, and that's called a mycelium. And so I often get questions about can you overharvest mushrooms? As long as and there's been studies that look at this over many years, finding no evidence that harvesting the fruiting body of the organism is detrimental to the rest of the organism, the mycelium mat, at all. So keep that in mind as you're out looking for mushrooms. So this mycelium, it's composed of single strands or hairs of fungal cells called hyphae. And the mycelium is usually hidden in the soil or wood or whatever substrate is growing on, and they secrete enzymes, digesting the organic matter, and then suck those juices back up. And when conditions are right, it's usually length of day, temperature, humidity. Here in Montana, we're pretty temperature and moisture limited. A typical mycelium will generate what we call a nugget of hyphae. It's called a primordium. And this is what will develop into a mushroom. And some of these primordia can live kind of semi-dormant in the substrate for many, many, many years. And the mushroom is designed to release the spores, okay? So real quick, I'm gonna recap some mushroom parts. As I might say some of these terms as I walk through the talk. But if we start at the bottom of this image, those what look like roots are the mycelium. You move up them, and you might see in your books the stem called a stipe or a stalk. Oftentimes there's some interesting features to note on the stem or the stalk, like a ring or an annulus. As you move up on the underside of the mushroom cap, you can have different structures where the spores are produced. Most mushrooms you're familiar with have gills, but if you know what a king bolete or the boletes are, they have pores. Many polypore fungi also have pores. And then as we get to the top of there, the cap, you'll see that called the pileus or pileus in some of your guidebooks. Okay, so the fungi in general have kind of three main life strategies for getting their food. And so the first group are parasitic fungi. We know what parasites do. They feed on living organisms. So many of your polypores or your conchs or shelf fungi are often causing heart rots or root rots, and they're parasitic on the trees they live on. The main group that you probably learned about in biology class are the saprophytic or saprophytes. And these are the ones that release their enzymes into the substrate and do most of our decaying out there. And then a big group are called mycorrhizals. And these are the fungi that form a beneficial relationship with many different species of plants. And I know you guys recently had Kathy Cripps give a mycorrhizal talk, so I'm not going to dive too deep into mycorrhizae, but for those that didn't see that talk, real quick, mycorrhizal, it means fungus root. And basically the fungal hyphae are smaller than your average plant root. And so they increase the surface area of plant roots that they're attached to. And so you get better absorption of water, nitrogen, and phosphorus. And the plant in turn is photosynthesizing and producing carbohydrates and sugar. And they're sending those down to the root system and interacting with this mycelium network. And this is kind of a grainy picture of what, these are some pine seedlings that were inoculated with one of the slipperyjack mushrooms, the Swillus species. And so you can see all that white fuzz is actually the mycelium, not the plant roots. The plant roots have a darker brownish or yellow color. And so you can see it's greatly expanded when you have a mycorrhizal partner. So something like 90% of all land plants, grasses, mosses even, many of our trees do as well, have a mycorrhizal partner. And a Douglas fir, there was a Douglas fir, I think it was in Oregon or coastal Oregon or Washington, and they did a survey where they went around and took soil cores and root samples, and they found upwards of 200 species of mycorrhizal fungi living in and on its roots. And so these mycorrhizal networks can connect different species of trees to other plants, and then they can exchange nutrients. And this has often been called the wood wide web or nature's internet. And so here's just another schematic showing you kind of how this works. There was a big study done, I think Kathy mentioned this, a big study done in the late 90s, where they injected radioactive markers into a tree, and they would follow it through using a Geiger counter, using a radioactive signature, follow where these nutrients, where the carbon actually went through these networks. And they found that you can see in the bottom of the schematic, I put some arrows for the direction of some of these nutrients are moving. And so you can see that fungus in the middle with the green network, that's its mycelium. The plant roots are kind of grayed out. And then you can see a saprophytic fungus there on the one side, it's not connected. And so depending, what these studies have found is that depending on the time of year, the seasons, what plant species are involved, the direction that these nutrients flow can change actually. And so it's a very dynamic system, and every day, the more we dig into it, the more we're discovering new connections. Okay, so back to these two groups of fungi, the two main groups of macrofungi, the Ascomycetes and the Basidiomycete. So what's the difference? The difference is how the spores are produced. And so Ascomycetes, the spores are produced in what we call an ascus. And just think of that as like a tube. Oftentimes there's 8 spores as depicted in this photograph. And when those spores are ripe, either there's a hole at the one end or a cap lifts off and the spores can shoot off. Versus Basidiomycetes, the spores are produced from a basidia, which are just like a little club, and the spores are out there just kind of dangling, and when they're ripe, they fall off and blow away. And here's just kind of a diversity slide of different spore types. So you may have heard of what we call LBMs, little brown mushrooms, and they may look, one little brown mushroom might look just like the other little brown mushroom you have in your hand. But when you do some microscopy, you look at the spores, the diversity is actually, it's pretty easy to tease many of these groups apart when you dig in there. Okay, before I jump into the first group, the Ascomycetes, I just want to take a step back. So you have this mushroom, right? It's designed to release spores. And most mushrooms release their spores in wind currents. But water, they can attach to bird feathers. Many invertebrates and animals eat them. It also spreads spores that way. But so you say a spore is mature, it releases from one mushroom, it floats away in the wind, it lands someplace. That spore will germinate into a hypha. And then that hypha will cruise through the substrate, and it has to meet up with a compatible mate, compatible sex of the same species. And they join, swap nuclei. then they become, they're haploid as, so they have one set of chromosome or one set of nuclei in the single hyphae, but when they join and have sex, they become diploid, and then they can form a fruiting body and make more spores. Now that's kind of a simplified view of it, because fungi don't just have two sexes like most mammals do, male and female. They are called mating types, and many Basidiomycetes have, I think the record is 25,000 different sexes. So yeah, think about that for a second. It's not just male or female. And why would that be beneficial? And it's basically the more mating types you have, the more likely you are to outcross. And so plant people are familiar with plants being able to self-fertilization versus outcrossing. Basically, for a mating type, or for when the hyphae come together and be able to mate, they have to be of different mating types. So they can't mate with their cells. So it's, if you think about having 20-some thousand different sexes or mates, you're almost always guaranteed to run into a mating type that you can join with. Okay.
If I could interrupt,
Okay, so this,
if I can interrupt just
Yeah.
briefly, Tim, there was just a question about the beautiful spores that you showed, and how much were those magnified?
yeah, so you can see some scale bars on there. So like if we look at this top one in the middle, I don't know if you can see my cursor, but 5 microns. The average spore is something in the order of 5 microns wide by 10 microns long. Some of the bigger spores are 400, 500 microns long. And you can see here, some of them are single celled, some have multi-cells, some are long and needle-like, some have color, some don't have color. And some of these spores are actually in the process of germinating. You can see some of them are starting to branch out. Like this one down here in the corner, you can see a hypha starting to germinate there.
Thank you.
You bet. So yeah, these are under the light microscope. They're quite tiny. This is at 1000x under the light microscope. So, but you can see when you go out collecting mushrooms, as I'll talk about a little bit later, is one of the key characters for distinguishing mushrooms in the field or after the fact is the spore color, right? And so you can make a spore print. And so when there's lots of spores, it's thick enough that you can see it on a piece of paper, but you can't see individual spores. Okay, the first group, the Ascomycetes, these are the cup fungi, right? And I kind of say to students in some of my classes, if it looks like an alien, it's probably a Ascomycete. If it looks like a mushroom, it's probably a Basidiomycete. But yeah, so they have this typical cup shape. The majority of them do. And if you were to slice through that cup, there would be hundreds or thousands of those ascus lined up, those tubes filled with spores. And changes in air pressure, moisture and stuff will cause those spores to be ejected. And so I'm an Ascomycete, I'm a cup fungus, right? I'm on the ground in the forest, say. There's always needles, there's always leaves, things raining down on me. And since I want to get my spores up into the air currents, this next slide shows that the cups, they're not just sessile on the ground, now they've grown a stalk or a stem, and they're starting to lift themselves above the moss or above the duff to get those spores up into the air currents. And then these two, actually, this purple cup is actually a group of Ascomycetes that are called hypogeous, and they basically do their life cycle underground. And so in order to find this purple crown, you have to actually lift a pile of duff off to see it, because it doesn't actually erupt on the surface. And so here in the Rocky Mountains, in any mountain range in the West here, we have a group of fungi that are called snowbank fungi. And so these are fungi that fruit right next to melting snowbanks high in the mountains all throughout the summer, as long as there's snow present. And this is one of the earliest members of the group. It's one of the first mushrooms you can see near melting snowbanks. It's called the orange peel mushroom. Nice, brilliant orange cups. And you can see here in this next photo, we have a snowbank down here in the right corner and these beautiful orange cups just inches away. So maybe the day before or 2 days before, that snowbank was covering this area. It melts back, you get a flush of water, sun hits it, soil temperature goes up. That's what's triggering these guys to fruit. We have something like, it's over 40 some species of snowbank fungi in Montana. So these guys are exclusively, it's a pretty pretty cool microhabitat, exclusively fruiting only near melting snowbanks. Here's a beautiful one. This is called the green stain, and you may have seen our pines get this blue or green stain to them. And you probably haven't maybe seen the fruiting bodies. It doesn't fruit that regularly, but you can often always find green stained or blue stained wood. And actually, when you work with, if you're a woodworker, it actually can make beautiful, beautiful pieces of art. And this guy's actually carried around by bark beetles and other insects. So as they're munching the wood and the bark there, they inadvertently carry spores to the next tree. Okay, right, so I'm a cup, I'm on the ground, then I grow a stalk, I lift up. Now I'm going to invert my cup, and instead of just being a single cup, I've now made many cups, right? And contorted ridges. And so basically, morels are a classic example of this. That surface area to be able to form and release spores is greatly increased when you get complex like this. And historically, we only had a handful of species of morels. We had, black fire morel, we had a gray or black natural morel. Those are two pictured in the middle. And then we had this river or yellow morel. I think it was in 2016, we did a big genetic study of the Morchella genus. And what was like a handful of species in North America went to 60 or 70 species. And so now in Montana, we have 6 or 7 different species of black fire morel. We have 2 or 3 species of gray natural morel. We have maybe 4 species of black natural morel. We still only have 1 yellow morel. This guy hasn't been split up. But if you have picked fire morels, you go into a burn area the year after a fire, and you often hear people, experienced pickers, say, "Oh yeah, I picked this flush or that flush." And basically, they used to be all lumped into one species, but these different flushes, basically, if you go in real early to a fire, you might find a specific species of black fire morel. But if you go a month later, it'll be a different species that's fruiting now. And so we didn't recognize those. They all look the same. Morels smell the same. They have the same texture. To me, they all taste the same. So it doesn't really matter if you're looking for food for the table, but the diversity is higher than we imagined previously. That's kind of what you want to see at the end of the day. Okay, so this is kind of sticking with this plan. We have a group called false morels. It's the same idea. They're contorted. They're still Ascomycetes. Sometimes they're saddle shaped. The one here in the middle, this is one of the snowbank false morels, and that can be quite big. It's a softball size, and you can see it there fruiting right through that snowbank. And kind of the classic way to distinguish between a true morel and a false morel is you slice the mushroom lengthwise, and all these ones pictured here will have like a pith, a white or fuzz inside, whereas a true morel will be hollow inside, and that holds up pretty well. Some of these species, there may be people on this talk be like, "Oh yeah, I've eaten this mushroom dozens of times," this one on the left, or these thimble morels here. The problem is they produce, and all mushrooms in the Morchellaceae family produce what's called monomethylhydrazine. And it's a very volatile chemical. It's actually one of the main ingredients in rocket or jet fuel. And it's a neurotoxin. And so if you inhale too much of it, it can cause brain damage. But since it's so volatile, it's easy to cook off. And so if you fry your mushrooms up, boil them in water, most of that monomethylhydrazine is cooked off. But you don't want to be over your pot breathing that in. You want to do that in a well-ventilated area. I don't recommend many of these mushrooms for the table because that's a subjective thing, whether or not your monomethylhydrazine is cooked off all the way or not. And usually, if you're finding these guys, there's morels just around the corner. So I don't recommend this group, the Gyromitras and Helvellas, for food. Elfin saddles, they're another closely related group to the morels. And you guys can see these guys are getting quite contorted. It's very interesting to see those pits in the stalk, in the stem of these guys. It's quite complex. And why would that be beneficial? Maybe I think there's a couple of reasons. The increased surface area makes it hold water and catch water better. But also I think it slows down insects and other critters that want to eat and chew on this, so they can get lost in these cavities and stuff. And they're trying to protect that fertile surface at the top, the surface that's producing the spores. So it's cool to keep your eye out for these kind of cool structures. Okay, so that was kind of briefly the some kind of major groups or some of the more showy Ascomycetes. As you saw, if you may have remembered earlier, the Ascomycetes, there are more Ascomycete species named currently than there are Basidiomycetes. And I show a slide towards the end that we'll talk more about that later. But that's kind of the section on Ascomycete. So hopefully I want you to recognize what a cup fungus can look like and how they differ from Basidiomycetes. And that's what we're going to start now, some groups of Basidiomycetes. And I'm going to go through based on spore color. So I'm going to start with white or light colored spores and kind of slowly move through to dark colored spores. And so I start with the Amanitas because I'll take a time out here. So we'll talk about mushroom poisoning. So let's say there are 100 mushroom poisonings in Montana every year. There aren't that. I don't think there's that many. There's not that many reported anyways. I would say something like 75% of those poisonings are not some toxin or chemical that the mushroom produces, right? So think of it like this. You made yourself a delicious turkey sandwich. You take that turkey sandwich out in the woods and you put it on a stump. You come back in 3, 4, 5 days. Are you going to eat that turkey sandwich? Probably not, right? Other bacteria, other fungi, insects, mice, things have come in, and it's no longer just a turkey sandwich. It's now a little ecosystem, right? So mushrooms have protein in them. They have water, some vitamins. There are always critters out there trying to eat them. And so most of those poisonings are not some chemical, like I said, it's some secondary, say, mold or bacterial infection. You're getting food poisoning. Basically, the common symptoms are gastrointestinal upset. And that's has nothing to do with anything the mushroom is producing. However, with the Amanitas, this group, I think it's like 3 of the top 5 deadliest poisonous mushrooms in the world. Three of those are Amanitas. They do produce a suite of chemicals that can basically digest your organs, liver and kidney. And it's a slow process. Usually, and there are fungi, you'll see in this talk, fungi, there's always an exception to any rule with fungi. But usually if you have symptoms pretty quickly after you consume a mushroom, you're probably going to be safe, right? It's probably a food poisoning. Whereas when you eat an Amanita, sometimes you don't get symptoms for a week or even 10 days. And then it might be another week or 10 days before you have liver or kidney failure. So keep your eye out for Amanitas. And how can you identify an Amanita? Amanitas have 2 very distinctive features that help separate them. The first is they have what's called a universal veil. And a universal veil is, just think of it as like an eggshell covering. And as that mushroom is developing underground, this covering, this universal veil, just protects all the parts that are inside developing. And so as that mushroom expands and starts to grow, the universal veil will oftentimes break up into these warts, like you see in these 2 photos here. And they often leave a bulb or a sac at the base of the mushroom where it looks like the mushroom erupted out of it. And this is the fly agaric, Amanita muscaria, and you can see the kind of a button mushroom here on the left showing just a little bit of red. Actually, there's a really nice yellow young button here on the picture on the right. And then they grow bigger, the cap expands, those warts start to spread out. And then as you get more rain or cold temperatures, the cap color can fade, these warts can wash off. And so when you're learning a new mushroom, it always helps to, collect multiple young ones, middle-aged ones, and old ones. And you don't want to slice your mushroom off. You want to dig it out, because oftentimes, if you were just to slice this Amanita right off at the base, you might miss that bulb, that part of that universal veil. That would be a key feature to help you identify that. Okay, so this is what's called the Western grisette. This is actually a delicious edible mushroom. It's an edible Amanita, but I don't recommend this for people because there are a couple similar mushrooms that could kill you. But you can see that the one that this fruiting body that I pulled out of the ground, you can see that brown cap kind of just bursting out of that egg. That's the universal veil. And you can see a little bit of flap of flesh left on the top of that cap. And so if actually I dig this out in the next photo, you can see, right? You can kind of imagine, push that brown cap back into the ground, reconnect that white patch to the other white patches down below. There's your little egg. That's your universal veil. I'll talk about puffballs later, but oftentimes people, when you collect a puffball, you want to slice through it to see if it's fresh. But you also, if you were to slice through an Amanita egg, you would see a mushroom shape in their developing. You won't see that in a puffball, but they have been confused in the past. Here's the booted Amanita. And I throw this one in there. It's called a booted Amanita because it's got that nice big bulb at the base, but it has a very, a good image of what's called the partial veil. So we just talked about a universal veil. Now we're talking about a partial veil. And a partial veil specifically just protects the gills or the pores. It's protecting just the fertile surface, the surface structures that are producing the spores. And oftentimes, as that mushroom cap expands, that partial veil will collapse and leave a nice little ring or a skirt on the stalk. And so Amanitas have both a universal veil and a partial veil. Here's the common grisette. And you can see that universal veil at the base here is actually very delicate, and it can wash away. When you pick it, it can crumble and you can miss it. So do pay attention to that when you're learning new mushrooms. Okay, so kind of got the deadly poisonous ones out of the way. There are a bunch of other poisonous mushrooms. I think there's something like 8,000 species of named Basidiomycetes in North America currently. And it's really only there's only 20 species that are considered deadly poisonous. And there's probably only 20 species, 25 species that would be considered a highly prized edible mushroom. And so if you can learn 20, 40 or 50 species, you're pretty safe. Well, what about the other 7,000 odd mushrooms? Well, a lot of them we just don't know. They're too small. And so, but most of them are probably not deadly. But yeah, so sorry. So this is the next group are the waxy caps. These also have white spores, but you'll see they're lacking the partial veil. They're lacking the universal veil. The waxy caps tend to be super colorful. And as their name suggests, even in dry weather, they have a sort of a waxy feel to them, both the gill surface and the cap surface and the stem. When you touch them, even when dry, they kind of have this wax paper, waxy feel to them. There are many non-poisonous waxy caps. Very few that are considered highly prized edibles. But these are many waxy caps are mycorrhizal with moss species and grass species. So a pretty cool group there. And we have something like 25 or 30 species of wax cap in Montana. And that was the genus Hygrocebi. There's another genus in the wax cap family called Hygrophorus, and they're also pretty colorful, but they're beefier mushrooms, bigger mushrooms. We have the yellow flaky wax cap up here on the left. We have the spruce wax cap, mycorrhizal with spruce. We have the purple wax cap here, and then this bright orange one. This is the larch waxy cap. It's a cool one because it's very common. You see it out anytime there's moisture, really, in mixed conifer forest. And it's an edible mushroom. And it's beautiful, but it produces butyric acid. If anybody knows what butyric acid is, it's the main ingredient in fake movie buttered popcorn. So it's kind of the smell of butter or rancid butter, actually. And so this one, you don't even have to add butter to the pan. You can just kind of dry stir fry it, and the whole house will smell like butter. Okay, so the next group are tricholomas, and this is a big group of mushrooms. And historically, if you were a generic looking white spored mushroom, you were thrown into the genus Tricholoma. This has now been split up, both on morphological studies and genetic studies, into many different genera. But here are a couple. This is a yellow one. This is called Man on Horseback. And I don't know where it got that name. This pink one is called the plum custard. Yeah, when you slice into it, it's yellow, but pink on the outside. This orange one is called the orange flag. It's Tricholoma focale. And take a time out here. So mushrooms can produce a whole suite of different odors, different smells. And that makes sense because many fungi are actually preyed upon by animals, mammals. They need to find them, sniff them out, and they help move their spores around. And so this is one of the mushrooms, and not everybody has the same power of sniffing ability as the next person, but this Tricholoma focale is an important one because I can smell this mushroom way before I even see it. Its smell is so strong it permeates through the whole forest oftentimes, and it smells like someone dumped a can of like bathroom cleaner, like citrus scented bathroom cleaner or something like that. I get excited when I smell that because this is an indicator species for me for finding the Matsutake, which is a Tricholoma. If you've ever heard of the white Matsutake. And this is another one that you can often smell as well before you walk up onto it. And it's, if you've never smelled a Matsutake, it's a smell like nothing else on the planet. It's a cross between like cinnamon and like red hots, like the candy red hots, and then it goes all the way over to like sweaty gym locker smell. So, you know, that might not sound delicious to someone who's never smelled a Matsutake, but it's a very unique smell. I mean, what I like to do with these is, you know, when you get these young buttons, you can just slice them into 3 or 4 or 5 thick slices on a piece of aluminum foil, sprinkle some soy sauce on there, wrap that foil into a ball and throw them in the freezer. And then when you want a little Matsutake morsel, you can just pull that foil ball out, put it in the oven or toaster oven. And then after you cook it, you can kind of just put a nice big thick of Matsutake on a cracker or a piece of toast. And that's the way I like to do it. However, I didn't find any Matsutakes this fall. So I'd like to hear if anybody stumbled upon these when they were out looking. And this is what you want to see, right? This one of the mushrooms towards the back there. It's kind of, you can see that pile of duff. This is one of the group of mushrooms we call the duff humpers. It's a fun name. Oftentimes, as we walk in through mossy or real, humus-rich forest, you'll see these mounds. And if you just go up and pull the mound off, you'll find a mushroom under there. It's a good skill to try to hone when you're looking for different edible mushrooms. And historically, this was actually one of the most expensive mushrooms out there, at least for North America, maybe second only to the European truffles. And the reason was the Japanese have a long history with this mushroom. They have another Matsutake. It's a different species, now recognized as a different species that grows in Japan. And kind of the best gift you could give a recently married couple was a matching button mushroom, one to the groom and one to the bride. And there are stories and of people selling matching pairs of button mushrooms for upwards of $5,000, $10,000 for 2 mushrooms. Kind of amazing. And the reason is there's a big Japanese market because historically, you know, these are perennial mushrooms. So many of our edible mushrooms the mycelium is underground doing its thing, connected to the mycelial network. And so you could come back to that same spot if conditions are right every year and pick more mushrooms. And so people had their patches, right? And somewhere along the line, someone discovered that if you rake your patch out, the following year you'll get way more fruiting bodies than you did the previous year. And why might that be? Well, you think about it, right? The mushroom is designed to release spores to start the next generation. Well, you disturb the mycelial network, right? That mycelium is like, oh gosh, my habitat is being destroyed. I'm going to take all my energy and do one last big fruiting so that I can get some progeny out there, right? And so they do that, and then they spend all their energy. Following year, they didn't find any mushrooms. Next year, no mushrooms. Basically, people were doing this, and they were inadvertently killing off their patches. And so then they started coming over to North America, and the market for Matsutakes kind of just went through the roof. It's stabilized now. It's not so crazy as it used to be, but they still fetch a pretty penny. Honey mushroom. You guys may have seen or heard of honey mushrooms. Honey, not because of the flavor, but because of the color. And this is a parasite. It's the genus Armillaria. We have many species of Armillaria or honey mushrooms across North America, but they always grow out of wood or out of roots. Ours are always on conifers. And this is a cool mushroom. They're edible, but they are very long-lived. And so you want to clean them well and cook them on high heat if you're going to eat these guys. And another cool fact about this is a contender for the largest terrestrial organism. And I think it's by both area covered and by mass, maybe not by mass. Maybe aspen trees are larger by mass. But anyways, there's a fairy ring of Armillaria on the border of Oregon and Washington that was a couple hundred miles across. And so they went out and collected samples all throughout this area. And it turned out it was all one genotype. It was all one organism. And then they carbon dated it, and they dated it to be around 1,400 years old. So not only they're old, but they're also quite big. There's another big fairy ring known of Armillaria in Michigan. I think it's 50 miles across. But foresters hate this mushroom. It causes the root rot. And so if you come into a forest with a lot of dead standing conifers, often that's a sign of armillaria root rot. Okay, so the next group we're still in the Basidiomycetes. We're still in white spored mushrooms. This is one of my favorite groups. These are tiny mushrooms. The genus is Mycena. They come in many different colors and really cool cap structures and shapes, but they're often, you'll see hundreds of them often on the forest floor, quite ubiquitous. They're saprophytic, they're not mycorrhizal, so they're digesting our pine needles and leaves and stuff like that. Very beautiful, very diverse group, many species. Kind of easy to identify a Mycena to genus. It gets a little bit harder to identify the species. But you can see we got a bunch of purple Mycenas, yellow, orange Mycenas, Mycenas that bleed red juice, Mycenas that bleed yellow juice, purple juice. But pretty common. Anytime there's moisture out, you can usually find Mycenas. I throw this one up. This is another member of our snowbank flora. This is the snowbank Mycena, Mycena overholtzii. And I throw this picture up there just because it's got a very unique field character where the mycelium actually climbs up the stem of the mushroom. And I've actually seen individuals where that mycelium makes it all the way up to the gills. This one fruits real early, right? The snowbanks. Okay, so the next group are the brittle gills, the genus Russula. These are ectomycorrhizal, ecto meaning they just attach to the roots, they don't actually penetrate into the roots. Very diverse. I think there's pushing 100 species of Russula in the Pacific Northwest. They come in all kinds of colors. They're kind of a medium-sized mushroom, no real distinctive features other than cool colors, like no ring on the stem and stuff like that, no structures on the cap. But they're very easy to identify to genus because when you pick one up and you break the stem in half, it snaps cleanly like a piece of chalk. And the reason that is because most, like I talked about the mycelium and the hyphae, they're long, the cells are very long, stringy, and hair-like. Well, Russula, it's actually a very early diverging group of Basidiomycete, so they diverged off early back in Earth history, way before the rest of the mushrooms did. And so they have a different cellular structure. Instead of having long stringy things, they have these cuboidal or square-shaped cells, and that's why they cleanly snap like bricks do. A nice clean plane there. But many yummy mushrooms in this group. Some have very spicy flavors. Some have very seafood-like flavors. And you'll see one of the most common ones we have around here is called the short-stemmed Russula. And there it is, right? It's one of the duff humpers. You could easily walk over this mushroom, but if you pull up that duff, it's this big, dense mushroom. And this is, if you pick this mushroom up and threw it at someone, it would hurt when it hit them. It's like they're very dense mushrooms. And this mushroom, it's edible. It's usually bitter, very tough. Definitely not a highly prized edible mushroom. But when it's still down in the ground as a duff humper, when it's starting to grow, sometimes it gets infected by another fungus, another unrelated fungus called Hypomyces, and it turns it into what we call the lobster mushroom. And this is probably my favorite mushroom to eat, favorite log mushroom to eat. And so that's a Russula brevis. That's this mushroom in the last picture that is infected by this secondary fungus, this Hypomyces. And it basically says, I'm this little microscopic fungus. I don't have the time or the energy to build a big fruiting body to release my spores. I'm going to take over your fruiting body, not let you produce your spores and produce my spores. That's what that orange color is it's a pyrannomycete. So it's got these little flask-shaped asci that shoot spores out. Delicious mushroom. Once you see the white Russula, it's not going to turn into a lobster. The lobster, the parasitization happens underground before you actually see the mushroom. But this can be quite common in years in Western Montana. And it tastes and looks just like lobster. Okay, the next group, this is still in the Basidiomycete, still with white spores. This is the milkcap group. They're in the Russula family, so they snap cleanly like chalk as well. But they have an additional feature that makes them easy to identify, and that's they bleed a latex. And so if you do have a latex allergy, you don't want to put these in your mouth. There you can see I sliced this mushroom, the underside of this mushroom, and that white stuff is latex bleeding. So a lot of characters can be gleaned from this. So the latex sometimes starts white and changes to a different color. Sometimes it starts one color and changes. Sometimes it stays white. But This guy's called the red hot milky. And this is, the flavor is described as being acrid. And if you've never tasted a mushroom that is described as acrid, it's spicy, but it's a different kind of spicy than capsaicin. It's almost, I've never actually dripped acid or battery acid on my tongue, but I imagine that's what it would feel like. And you just take a little, maybe a tenth of one of these caps into a stir fry, and that's enough to give it a pretty hot, potent flavor. Okay, so that was kind of some major light-spored mushroom groups that are common in Montana. So now I'm going to shift to darker spore prints, and I'm going to start with black or purple black spore prints. And this is a group that we call the stropharioids. This one is, I throw this one up there because it's got some interesting features. This is called the questionable stropharia. And you remember we looked at that Amanita and it had that skirt, that partial veil, that skirt on the stalk. That's what happens with the majority of mushrooms. When that cap expands, that partial veil collapses onto the stem. Whereas this stropharia here does the opposite. When that cap expands, the partial veil stays attached to the cap margin, not to the stem. So you get these nice little flaps of tissue hanging down. And so it gives it a very distinctive look. It's very easy to identify. And then if you look closely at the gills here on this mushroom that's turned on its side, you can kind of, I don't know how it shows up on your screen, but you can kind of see a purple dark color coming to it. So you can infer without doing a spore print or without looking at spores under the microscope that this will have a purple or dark colored spore print. Stropharias are interesting for another reason. So a whole group of fungi have independently evolved the ability to eat insects or worms and nematodes and other invertebrates in the soil. And they've done this they can do this many different ways. But Stropharia figured have do it this way. So on the mycelium, they have what this structure would called acanthocytes. And just imagine like a microscopic jack or like a little ball with all these little needles sticking out of it. And they're spaced out every so often along the mycelium. And so you have this giant mycelial network with these, like, I kind of visualize them as like landmines, right? And so if you're a worm or a nematode cruising through the soil, coming in to say, eat this mushroom, you will rub up against that stuff and those needles will poke into you and all your juices will flow out and the mushroom mycelium will suck that up. And so this, all Stropharia have the ability to do that. And I throw this picture up there because this is Stropharia, the hornman Stropharia. It's a fairly regular deep forest, old forest mushroom. But I took this picture 10 a.m. one morning, and then I came back 24 hours later, 10 a.m. the next morning, and this is the same cluster of mushrooms. So people always ask me, how long do mushrooms last? How long do they're up for? Some of the shortest ones are hours, 3 hours. The mushroom shoots up, releases its spores, and withers back down in hours. The record holder for the oldest individual fruiting body is a 600-year-old polypore or conch living on an 800-year-old Doug fir. So those are kind of the extremes of how long a fruiting body can be up there. But you can see this was only 24 hours from when, so it was probably this had probably been up for 24 hours. And then 24 hours later, you can already see that purple or brownish color underneath in the center here. That's the spore print from the ones above it, raining down on the cap below it. But they're already starting to do their thing. They're releasing spores. They're starting to wither a little bit. So, on average, most mushrooms are up for days, maybe a week max. And you'll get cases where, like that armillaria can be up for a couple of weeks. Some chanterelles can be up for almost a month. Inky caps, we just kind of finished shaggy mane season. If you drive around any forest road in Western Montana or anywhere across the state, really, you can often find shaggy manes. I love this mushroom. It's called an inky cap because they have keyed into flies and other insects to release their spores. And so they auto-digest into a black inky mess. And when flies and insects come and land on it, that inky mess sticks to their feet and they carry the spores around. And so after you chop this mushroom, if you're more than 2 hours away from cooking them up, and you got to drive or you put them in your car, they're going to turn into a black liquid goop before you even get home. So you got to be quick with this one. But a word of caution. So many mushrooms have the ability to bioaccumulate heavy metals from their environment. And this shaggy mane has been shown it's kind of the super bioaccumulator. And so you don't really want to eat this mushroom, say, in a mine waste site, or there's some evidence even next to asphalt roads where you're getting rain leaching off petrochemicals into the soil. They actually use this mushroom, it can use this mushroom to clean up toxic areas, like they used it in Chernobyl even. And so you go in and you pick the fruiting bodies and put them in buckets and basically incinerate that. So it's just a way to collect quantities of heavy metals out of the soil kind of passively. Okay, so the next group, so we kind of left the dark or black spored mushrooms, and we're now into brown spored mushrooms. And this group, the webcaps, it's the genus Cortinaris. It's a very important group of mushrooms for a couple reasons. They're ectomycorrhizal. So if you see a lot of Cortinaris out there, you can kind of infer that if the Cortinaris are happy, the trees are probably happy. Not many mushrooms in this group are food. There's quite a few poisonous ones, and there's many undescribed species, and many that we don't even know what kind of chemicals they can produce or don't produce. And we don't know the ecology of a lot of these things either. So it's the most common mushroom you're going to find out there across the globe. There are more species of Cortinaris than any other Basidiomycete group. Very hard to get to species level, but very easy to identify to genus, because they have, as the name suggests, they have a cortina. And a cortina is just a specialized partial veil. Remember, the partial veil protects the fertile surface. And so here I sliced this Cortinaris in half, and its timing was a little off, right? The partial veil hadn't broken open yet before the gills started releasing spores. So you see that cinnamon brown deposit at the top of the cobwebs there. And so it's a very distinct feature. If you have a cobwebby partial veil, you know you have a Cortinaris. Okay, next group. We kind of went through all the gilled Basidiomycetes. So they had gills, right? They had these, when you flip up the cap, they have these blade-like radial pattern of gills, right? And so if you were to look at a gill on side view, there'll be basidia sticking out either side, and on the end of those basidia are the spores dangling out. Well, the next group are the group of mushrooms that don't have gills, they have pores. That's often generally referred to as the bolete family or bolete group. This is the king bolete, the porcini, the steinpilz. This is a delicious edible mushroom. And one of the key characters for it is it's got this net-like pattern on the stem. If you can see that on that big one there, it's like almost someone imprinted a net onto the stem. Lexinum is another genus. These are the scaberstock or ref stem boletes. Many of these are actually poisonous or cause gastrointestinal upset. Very patriotic mushroom, red, white, and blue there. But you can see those glands on the stem there. The slippery jacks, these are also in the bolete family. We have many. This is a great group of mushrooms for the table. There are no deadly poisonous slippery jacks, and they're fairly easy to identify to genus. We have the short-stemmed slippery jack up there. We have Suillus corvelii, or the tamarack jack here, this real goopy red one. Same picture here, the tamarack jack. We have the western painted Suillus down here. Very yummy mushrooms. Some people don't like the texture. They can cook up fairly slimy. There are different ways to prepare them so you don't get the sliminess, but I think they have a great flavor. This is the hollow-footed slippery jack. So I show this one in there. When I slice this one in half, you can still see the partial veil is still attached. But its other unique characters, no matter if it's young or old, you slice through it, it always has a hollow stem, like that, a cavity inside of it. So very cool, easy to recognize field character. The blue staining slippery jack. So when you bruise or cut this one, I don't know if it shows up there, but it almost instantly stains blue. And here you can see the pores, right? You can see those the holes there on the lower surface, but you can see these almost like tubes going up. So inside those tubes, they're just coated with basidia dropping spores, raining down spores. So the next group, right? They're not really gills, they're not really pores. It's kind of something in between. They call them ridges. This is one of our golden chanterelles. We have a couple species of yellow chanterelles in Montana. This is our big beefy one called the Cascade chanterelle. This is called the rainbow chanterelle. So there are still chanterelles out right now. They actually don't mind a cold as much as some other mushrooms, so they can handle the cold quite well. And they're actually never really that buggy either. So, and they're long-lived. If you find a patch of chanterelles, you can revisit that patch for decades, seems like. This rainbow chanterelle is mycorrhizal with spruce. So I tend to find it right as you start to leave the lodgepole pine and into the spruce fir zone. The white chanterelle, the blue chanterelle. This might actually be a truly rare mushroom. I don't know. It can be locally abundant, but I never see it in the same place year after year. But it's a gorgeous mushroom. It actually looks like a pile of bear scat when you walk up on it first. There it is again. Okay, so the next group, this is the Bluetooth. So we're not gills, we're not pores, we're not ridges. But if we flip this little, it's not that little size, a tennis ball, flip it over, it's covered with spines. And so instead of being a tube with the spores on the inside, the spores are on the outside of these spines, waiting to mature and be released. You slice through this mushroom, and you can see the spines dangling down here. But you also notice these rings, right? You see these kind of bluish gray rings going out. Those are growth rings, just like in tree rings, except they don't represent a year. They represent start and stop of growing conditions. So, moisture is not always present, right? You get a rainstorm, you have moisture in the area, you can grow a little bit, things dry out, you stop growing. Next rainstorm, you grow a little bit more. And so they show, if you can kind of age them. I think this one's probably 4 years old. There's kind of a few packages of growth rings, like you can see maybe 10 rings in a season or something like that. So you can slice these open and kind of get a sense for what the rainfall in that area was like in the past few years. It's pretty cool that way. Here's the orange tooth, and it's mushrooms are 90% water, right? So sometimes there's too much water and they exude a juice. This one actually has a little bit of sugars in it, so it does attract flies and insects too, so they can come and slurp that up and spread spores. The hedgehog, I love this mushroom too. This is one of my top 5. This one smells and tastes like brown sugar when you cook it up, but it has a kind of classic mushroom shape, but it's got spines on the underside. This is called the shingled hedgehog. This guy can be as big as a dinner plate. It's got very big scales on the top, so pretty easy one to identify. It's edible. We have probably 6 or 7 species of Sarcodon in Montana. So there's always there's fungi out there eating everything that our forest is throwing at them. So we got lots of pine needles to eat. We got lots of, in this case, pine cones, fir cones. There are many fungi that specialized on these different substrates. And this is one, it's called the conifer cone tooth. It's really tiny mushroom. It's probably only the size of your pinky. But when you take a close-up picture there, it's just got this nice kidney-shaped cap with lots of little teeth on it. So now I don't even have a stem and a cap. I'm just all spines or all teeth. This one's called, you often hear it called bear's tooth or bear's head. I call it conifer icicle. We have 4 species in Montana, 2 that grow on conifers, 2 that grow on cottonwoods. And these can be quite large. I think one of the biggest specimens seems like 30 or 35 pound individual. And I love this mushroom too. You can kind of what we do is we just make pizzas, and you kind of just crumble it on top of the pizza and bake it in the oven, and just add a different texture, flavor to your pizzas. Also really easy to grow. So you want to cultivate this mushroom, it's quite easy to grow. Wood ear, right? So now we're into the jelly fungi. So these don't have a real defining structure for releasing spores. They kind of can release spores all across their surface. And they are kind of flabby and jelly-like. If you've ever had sweet and sour soup, or these brown, stringy, crunchy things, that's this mushroom, the wood ear. Grows on downed conifer logs, mostly picea or spruce and fir, still with the bark on it. Witch's butter, non-poisonous, but if you like eating snot, go for it. The tooth jelly, right? So I'm a jelly fungus, but I also now produce spines. Next group are the puffballs. And so I don't have my spores on the outside. I have my spores on the inside now, right? And they're not on special structures. They're just in one giant spore mass. All puffballs are edible when they're young and fresh. You slice through them. If they're white, they're good to go. If they're any kind of color, like this one up here in the right corner, it's starting to see that yellow and crack and open. It's overripe. It's starting to release its spores. The polypores, right? So these are the shelf fungi, or your conchs. They have pores on the underside, but they grow directly out of wood or standing or dead wood. turkey tail here down on the left. This one actually has a lot of strong medicinal properties. Looks like it's going to be a good one for fighting bone cancers. The sulfur shelf, also known as chicken of the woods. I think this was like, we harvested maybe 75 pounds of mushroom off this one over one season. Here it is, a little younger and fresh. It can get quite woody, so you want to collect young, fresh ones. Corals, right? So instead of my spines hanging down, my spines are going up. We have, I think there's 60 or 70 species of the genus Romeria in the Pacific Northwest. Here's just showing the diversity of some of those. Many of our corals are edible. You hear lots of stories of causing gastrointestinal upset. And like I said before, these, if you look at these, they're pretty complex, right? They're hard to clean. Lots of insects, other fungi, other molds and stuff they find these a nice home to grow on. So cook them on high heat, wash them well if you're trying them new. The cauliflower mushroom. I think this is another truly rare fungus in the state. It's a root rot, usually a root rot on larch, I find, but huge basketball-sized clusters. It's a delicious edible mushroom. It's like a pile of egg noodles. Kind of has a taste like egg noodles too. It's kind of, the texture like egg noodles. Club fungi. So these are not complex branching like the corals were. They're just kind of individual fingers. Candy club on the right here tastes like brown sugar when you cook it up. Really yummy mushroom. Bugs often get to it before I do, though. The strap corals, these are just kind of, when you see them, there's hundreds of them scattered throughout the forest floor. The bird's nest. So this is kind of like, think of these as like, they make a cup. They're a Basidiomycete, but they have little puff balls inside of them, what we call the eggs. And so when raindrops hit this, they splash these little spore masses out, and they'll disintegrate into a pile of spores. And here, I think I have a picture of it here close up. You can see the one on the right, is young, hasn't even opened. One on the middle right, it's just starting to open, and then the two on the left have already ejected their eggs. Common in wood-chipped gardens and stuff like that. Here's the jellied bird's nest. Same idea, but instead of shooting those spore masses out through raindrops, when it gets wet, there's a jelly inside that just swells up, and the thing just kind of oozes out and erupts. Okay, so that was kind of just a brief overview of some of the major groups of fungi that you're likely to come across in Montana. Do know that there are many, many other groups that I didn't talk about. But I'm just going to give you some numbers here. So how many species of fungi do we have in Montana? The current, the thought is for every vascular plant in an area, you have anywhere from 6 to 10 species of fungi. And so we know we have about 2,500 species of vascular plant in the state. So does that mean we have 15,000 species of fungi if we take the conservative estimate of 1 to 6? I'm going to say probably something close to that. I'm working on the checklist for fungi for the state with the heritage program. And our draft list right now is just pushing just under 2,000 species on the list. So we have a long way to go. Lots of unknowns out there. Just some of our neighbors, some numbers, and this is just macrofungi. So 1,600 from BC, 2,400 from Washington State, another 2,000 from Oregon, 1,600 from Idaho, about 4,200 macrofungi total for the Pacific Northwest currently. So here's a slide from the Kew Botanical Gardens put out the State of the World's Fungi a few years ago. And this is, we have year on the bottom. On the left, we have the number of new species described as science that year. And so you can see we've kind of just been steadily describing new species every year, but recently, mostly because of genetics and genomics, those numbers have kind of gone up. So you can see in 2016, we had over 2,500 new species of fungi described as science that year. Lots of work to be done, lots of new species everywhere you look. Okay, one more slide here. I put this together. On the left, there are kind of just like the 20 common edible mushrooms in the state by species there, and then the calendar year across the top. And then the color coded, so dark green means that's when they're most likely to be found, light green, less likely. And so you can see our edible mushrooms are highly skewed towards the fall season. And in Montana, we really have two distinct seasons. We have a spring season and a fall season. Usually gets too hot and dry in midsummer for much to go on fruiting wise, except unless you go way high up in elevation. And I didn't talk about lichens at all in this talk, but not all fungi are as simple as they seem. There are many what we call Basidiomycete. So these are, you look at this fungus and be like, yeah, that looks like a mushroom. But if you were to follow its hyphae down, its hyphae are basically, it's like a glove. Here's a ball of algae, and the hyphae come around and kind of encapsulate that algae, and they're stealing or sharing photosynthetes from the algae. So it's, oftentimes lichens are described as just fungi that have discovered agriculture. And this is just an example of a Basidiomycete that has been lichenized. All right, guys, that's all I have for tonight. If you got any take pictures of mushrooms you want to get a name on, you want to know if it's safe to play with, send me a picture at my email here, timothybwheeler@gmail.com. Yeah, please check out my website. A lot of these photos you'll find up there. We have there's the Western Montana Mycological Association, this group in Western Montana. Check them out. They do a couple meetings every year. If you're really proficient with using keys to key out plants or fungi, the Pacific Northwest Key Council has, like, if you have a bolete, you have a whatever chanterelle, you can go and look at a key to all the chanterelles or a key to all the boletes for the Pacific Northwest. Really good resource. And then if you want to get into lichens a little bit, Ways of Enlightenment is a good group of folks publishing about lichens. So thanks, everybody.
Great. Wow, Tim. I know everyone's joining me from the couch with a big round of applause, and we thank you for all your work and for sharing it with us in such a great way. And I know I'm not the only one who's very glad that we have a recording to refer to your beautiful pictures and the identification. It's just wonderful. Questions are rolling in, so I think I can start fielding those to you. The first one was when we were looking at the Amanitas. Stella was wondering if you'd ever seen Amanita muscaria in Montana.
Oh, yes, definitely. And so it's mycorrhizal with pines. And we have at least two, I think they're currently subspecies, but we have at least two common species of Amanita muscaria. One that's more yellow. It's called flavovulvata. It has a yellow partial veil, more yellowy orange in the cap. And then we have the one that's the kind of a classic dark red that I showed some pictures of. Currently, they're both the same species, but there's some recent genetic papers that argue that these things should be species. So yeah, it's not as common as, say, if you go on the West Coast or like some places in the country where there are pine plantations where you can go out and just be carpeted with red amanitas.
Yeah. And Robin was, she'd always wondered about the Amanita warts and how they were formed. So she was grateful to learn about the veil and a little bit about the organism there. All right. Regarding the lobster, how can you tell what species is being parasitized by the lobster? Does it have to be examined in a lab?
No. The good thing is that the Hypomyces fungus that parasitize the russula, they're very species specific. So Hypomyces lactifluum almost only parasitizes that white russula. Now there's another Hypomyces that parasitizes only boletes. There's one that only parasitizes a different species, one of the milk caps. And they're different colors. So if you feel comfortable recognizing that orange color, the big size, there's nothing else you're going to run into that looks like that.
Great. Karen asks, what's the evolutionary advantage from producing a highly lethal apple like the Amanitas do?
Yeah, so the mushroom is designed to release spores, right? And think about it, right? You're filled with water. You can take a week to grow before you mature to release your spores. Well, you don't want to get eaten right out the gate, right? Before you produce your spores. And so the advantage is that predation is down, right? Because you eat this mushroom, the Amanita, many of the Amanitas, well, not many, but the fly agaric particularly is considered a hallucinogenic mushroom. And so if you're an insect, we don't know that they actually have effects like this, but you can imagine that You're cruising, you're eating it. It's going to slow you down, make your brain fuzzy. You're not going to be as efficient as eating the mushroom parts. So you basically have more time to release more spores.
Great. Barbara's curious, what gives each mushroom their distinctive taste?
Yeah, that is a great question. Biochemically, I don't know. That is not a rabbit hole that I've gone down yet. But there are a whole suite of secondary metabolites, smells, chemicals that mushrooms can produce. And yeah, I can't really say anything more specific than that, but the odors are very, very diverse. And you can train your nose to start to recognize these smells. And there are names for some of the smells. But smell is kind of weird. It's a weird sense, right? I think we underutilize as humans. I think we underutilize our smell as a sense. And so when you pick a mushroom up and you smell it, to me, it's always like, it smells like this, because you're like, it reminds me of something else, right? But you don't necessarily have a name for that smell, even though it's unique. It's always comparative, right? And so it's just a weird relationship that I have with fungi. I think I have a strong smell sniffer. And so when I come out in the woods, I pick a mushroom up. The first thing I do is take a big sniff of it. Second thing I do is I take a bite out of it. You're like, whoa, you're not supposed to eat raw mushrooms, poisonous mushroom. Even the most deadly mushroom, you can chew on it. You get the taste as long as you spit it out, right? It's only when you ingest it. And you have to ingest a decent amount to reach the toxic levels for most of these things. So you can touch them all day long. You can cut them open. You can smell them. You can taste them. Not going to kill you. Not going to harm you. So.
All right. Vicky's wondering if you have a feel for what % of the Basidiomycetes occur east of the divide.
It's a great question. East of the divide is way less studied than west of the divide for fungi. And you can imagine like kind of the first cut would be like, I'm an ectomycorrhizal with spruce, right? You're not going to find me anywhere that spruce isn't growing, right? So once you get way out into the prairie, into the plains, you're obviously not going to find all these mycorrhizals with spruce. I would say something probably like 30 to 40% of the mushrooms we find west of the divide, you could find east of the divide, and they're mostly going to be these saprophytes, right? Saprotrophs, that they can basically digest any type of leaf litter, etc., stuff like that.
And I don't know if she knows something we don't know, but Andrea is asking, when is your book on fungi coming out?
Yeah, so Kathy Cripps and I are working on a Montana mushroom book, and we are doing it by family. So kind of different than most field guides. And so we've worked through, I don't know, a dozen or so more, 20 families of mushrooms. We have the write-ups, pictures, stuff like that, but we're kind of just chugging along. I don't want to give a date. It'll be done when we think it's perfect.
Sounds good. And do you have any special techniques or equipment to capture these mushrooms in photographs?
Yeah, so a lot of I do what's called focus stacking. And so basically it's a technique where you take at shallow depth of field, you take one picture and then you take one a little deeper and one a little deeper, and then you stack them all so that your image is sharp from front to back. Because that becomes an issue. The more magnification, the higher magnification you're taking a picture at, the less depth of field you have. And so, and these things are 3-dimensional, and so you want to be able to see all the structures. And so I can do that in the field. So I have a camera on a tripod and take a photo, and I can do that manually where I just kind of change the focus a little bit. And so like a field photo maybe only has 4 or 5 or 10 photos stacked, but I oftentimes, for all the small stuff, I bring those into I have a lab at home, a studio set up with artificial lighting, camera on a rail system, a computer system. And I program the first picture and the last picture, and then I can walk away and it'll take pictures successfully through that image. And say like, if I'm at like super magnification with a complex mushroom, it could be 400 pictures that are stacked into one image. And so that's how I get them really sharp and crisp. And then you just got to play around with lighting to get the right color. And that can be done many different ways.
All right. Noah wants to know, what category does chaga fall into? Do we have chaga in Flathead Valley? I don't know if I'm saying that right.
Yeah, chaga. So chaga is a polypore. It's a conch or shell fungus. It's a parasite. It only grows on birches. There are records, or I don't know if there's any records, but there are collections and sightings of this thing. I have never seen an actual voucher of chaga collected in Montana, but it is assumed to be present here. Not very common. I know people. There are many chaga lookalikes. It kind of looks like this black crusty, like if you have a picture of what aspen bark looks like, how there's some kind of those black scars and stuff like that. That's what chaga kind of looks like. It just looks like a black scar on the tree. When you break it open, it's got a nice orange color on the inside, which helps. But people make tea out of this, and it's got some medicinal properties. Not common, not abundant here in Montana.
A number of people have been commenting on the art that's behind you, and someone asked, does the art represent part of a mushroom? And you better tell us a little bit about that.
Yeah, no, this is a lichen. This is a metal print of mine, and it's this is the lichen Caloplaca biaterina, and it grows on limestone. This is growing on a limestone cliff. This photo was taken down by Dylan. And it's just two individuals. And I don't know if you can see, but there's got these radiating lobes, right? And then in the center, there are all these little circles. The circles, just think of them as little cups, right? That's the fruiting body of this lichen, and they're shooting spores. The orange color is a chemical a secondary metabolite that the lichen produces called xanthoric acid. And it does a few things. It protects these things from UV. So they're often growing on really full sun, sun-blasted rocks and stuff like that, so they need protection from UV. But they're also bitter, and sometimes they're toxic to invertebrates, so it slows down. It's anti-herbivory as well. Yeah, it's a beautiful orange mushroom. There's quite a few. We have dozens, maybe 30 or 40 species of mushroom that are orange like that look very similar.
Great. Alexa is asking, are Matsutakis associated with Allotropa virgata, which formerly was a U.S. Forest Service sensitive species?
Yeah, so there's this, right, so there's a whole suite of what are called microtrophic plants, right? These are plants that don't produce chlorophyll, so they don't photosynthesize. So your plant that doesn't photosynthesize, how do you get your food, right? A lot of these plants are tapping into this mycorrhizal network, right? And they're stealing, borrowing, sharing sugars from that system, from that network. Yeah, and so Allotropa is an indicator species for Matsutake habitat. Now, I don't know specifically that Allotropa is connected to Matsutake mycelium. That's a question we have to do some research. I don't know that connection has been made, but yeah.
All right. And Andrew Smith has shared in the chat a link about some of the biochemistry about the distinctive odors for some
Thanks, Andrew.
people that are interested. And we're getting lots and lots of thank yous and kudos for such a great talk here. And is there anyone who would like to unmute themselves and make a comment or ask a question of Tim? You could feel free to do that now if you'd like.
Hello.
Go
Hello.
ahead. Diane?
Yeah. I live down in Dayton, and I lost about 15 acres of forest, mostly ponderosa pine, this July in the Elmo fire. And I know nothing about mushrooms, and I want to get started learning as much as I possibly can and what I should expect to pop up this next spring.
Yeah, so you said it burned. What kind of trees were they again?
Mostly ponderosa pine.
Yeah. So yeah, the morels will pop up the year after a burn. Usually the later in the season the burn is, the better the resulting fruiting is. So if you had a fire, say you have a fire in mid-August versus a fire in late October or something, the one in late October would produce more mushrooms the following year. Yeah, so there's a whole, I can go into a burn area and look at the species of fungi that are present. And up to about 10 years post-burn, I can tell what year we're in based on the fungi that I see. So morels, you only see the year after. Second year, often you see a lot of these foliotas, species of foliota. And you can do this to about 10 years post-fire. And so, yeah, and ponderosa pine tends to be a drier, most ponderosa pine sites are drier, right? The deeper your soil is, the more organics in the soil, the more moisture it's going to hold, right? Because if you burn off all the trees, you're now exposed to more sun, right? More wind. It's going to dry out even more quickly than it did historically. So,
Right.
yeah, it depends on how much moisture you get. That's going to be the major factor, depending on what you get. But you could definitely find morels there. There's no reason you wouldn't find morels next spring.
Okay. Well, I've got to learn everything I can, so I've got to start in the basics. So, yeah. Thank you.
Yeah.
All right. Well, with that, I'm going to stop